Hi, welcome to our symposium on simultaneous MR-PET imaging, recent developments and future directions. The objective of our symposium is to provide you with an introduction to simultaneous MR-PET and its application for studying human brain function. We'll provide a brief introduction to the dual modality technique, as well as opportunities and challenges of the method. In these first two presentations, we'll discuss simultaneous functional MRI and functional PET imaging to measure human brain function. In the third presentation, we'll hear about receptor imaging using MR-PET, and in the last presentation, we'll hear about trimodal EEG, fMRI, PET imaging and future directions at high field strengths. So why integrate MRI and PET? It's sometimes been said that a fully integrated MR-PET system was a holy grail of imaging technology. But why? From a neuroimaging perspective, MRI and PET have complementary strengths and weaknesses. So while MRI provides excellent spatial resolution, the spatial resolution of PET has a natural limit. On the other hand, PET imaging is remarkably flexible in the range of molecular targets that we can image, providing the capacity to measure different physiological properties of energy metabolism, neurotransmitter dynamics, protein, inflammatory markers, and so on. So from the perspective of functional neuroimaging, some of the markers of neuronal activity can actually be physiologically closer to the process of interest than the MRI signals. The first concept studies for combined MR-PET were performed in 1996. The full integration of PET and MRI hardware into a single system was a considerable technical challenge. However, many of those challenges have been addressed sufficiently so as to allow for commercial systems to become available by 2010. By 2019, there were 70 such integrated systems available worldwide. And an alternative to the fully integrated system is the brain PET insert, which involves inserting PET hardware into an existing MR system. And John will speak more about this in the last presentation. There are a number of experimental approaches to simultaneous MR-PET neuroimaging, one which primarily relies upon the spatial registration that's offered by the dual modality system. So for example, exploiting the anatomical sensitivity of MRI to correct artifacts like partial volume error, or to identify molecular activity with fine spatial specificity. The other approach aims to exploit the spatial and temporal registration of the simultaneous acquisition. So for example, to time lock functional MRI and PET dynamics. In 2018, we called this synergistic imaging. And while much more work needs to be done to truly synergize MR-PET, it's this type of spatial and time-locked MR-PET studies that we'll discuss most in this research. So what are the opportunities and challenges that we face with MR-PET at the moment? All imaging modalities have their challenges, and some of these are addressed by the dual modality system. Partial volume error is a significant issue for PET, which MRI is particularly well suited to address. The partial volume error is one of the primary limiting factors for the quantification of the PET signal and is a particular issue for small brain structures. The intrinsic registration between simultaneously acquired structural MRI and PET optimizes anatomical-based partial volume correction routines. MR-PET partial volume correction actually even performs better than the standard in PET-CT, since PET and CT are actually acquired sequentially within that system and not simultaneously. And so movement between the scans is possible and can impact registration. On the other hand, PET attenuation correction is more challenging in MR-PET than it is in PET-CT. Accurate localization in PET depends on accurate detection of the annihilation event, which is impacted by attenuation. Attenuation occurs when photons move through biological tissue or scanner hardware before it's detected. And so attenuation therefore leads to a loss of signal, but it also can lead to localization errors in the form of scatter. In PET-CT, the CT scans can be used to form an accurate attenuation map. However, this is not possible in MR-PET because certain tissue classes like bone can be difficult to distinguish from air using standard anatomical routines. Furthermore, attenuation is actually a more serious issue in MR-PET than it is in PET-CT because it requires the presence of additional hardware for MRI, such as RF coils and positioning foam within the field of view. MR-based attenuation correction remains an area of active research. However, possibly the most common method at the moment is to use ultra-short TE images, as shown here. Here's an image from our group from a few years ago, which is useful to see the types of error that some of the routine MR attenuation correction procedures provide. So while there's no standard approach yet to MR attenuation correction, the community has leaned towards these Ute-based approaches, with vendors providing these methods in their suites. So in summary, the development of simultaneous MRI-PET hardware took many years, and many of the technical challenges, particularly with regards to hardware, have now been addressed. This means we can start taking advantage of the many opportunities simultaneous MR-PET provides for studying human brain function. However, it's important to understand the challenges that still remain with the hybrid imaging approach, and these methodological challenges influence the data quality and the post-processing procedures that must be carried out to ensure data accuracy. One application of simultaneous MR-PET, which we'll speak at length about today, is a combination of functional MRI with FDG PET imaging of neuronal glucose uptake. Glucose is one of the primary sources of energy in the brain, and regional glucose uptake primarily reflects activity at the excitatory synapses. So glucose uptake can be considered an index of neuronal activity that is physiologically closer to the biological process of interest than the bold response. FDG PET of the brain is routinely acquired in both the clinic and in research settings. So the standard approach to FDG PET uses the bolus method, where the entire dose of FDG is administered all at once. Subjects then rest in a quiet room during the uptake period and are then scanned for 10 to 30 minutes. So because PET is an accumulative signal, the recorded signal therefore reflects the integral of neural activity that occurred across both the uptake and the scan periods. As such, the recorded signal is not actually specific to the cognitive processes or neuronal activity that has just occurred during the scan process. Lastly, standard FDG PET acquisitions are usually what we call static, in the sense that a single image represents the cerebral metabolic rate of glucose over the entire scan period, which can last from anything from 10 to 30 minutes. One method that's been developed at the same time as simultaneous MR-PET is the so-called functional FDG PET method. And it's called functional PET or FPET to highlight the similarities with fMRI. However, it's important to note that you could also use this method in other approaches using PET-CT. FPET uses the constant infusion technique, where FDG is administered by infusion over the course of the scan period. And the goal here is to maintain a constant plasma supply of radioactive glucose so that dynamic changes in glucose uptake in response to a task or stimulus can be measured. And I won't spend a great deal of time on the acquisition procedure here, but if you're interested in how to acquire this sort of data, you might want to check out our Journal of Visualized Experiments video article where we demonstrated the procedure. In 2014, the MGH group published a proof of concept study which demonstrated the potential of the FPET method. They showed that a constant plasma supply of FDG allows measurement of dynamic changes in glucose uptake in response to a checkerboard stimulus. And here you can see the model fit within the visual cortex in response to alternating blocks of flickering checkerboard, which they measured with FDG PET using a 1-minute frame duration. In this study, they didn't acquire simultaneous fMRI, just simultaneous structural MRI. So this landmark study set the stage for a number of studies that have continued to develop and refine the FPET methodology. A lot more work is focused on the additional information that can be gained by acquiring FPET simultaneously with fMRI. And Andreas will speak a lot more about this area, particularly with regards to modelling the data. I'm going to focus more on the application of FPET and fMRI in the resting state. One question that's been explored since the Verleen paper is how we can improve the temporal resolution from the 1 minute reported in that original paper. The Vienna group published this nice paper in 2018 and focused on manipulating the experimental design and task duration. On the basis of this study, they concluded that at a task duration of 5 minutes, 30-second frame rate is optimal. In a forthcoming paper, we've examined whether combining a bolus and constant infusion can provide a better temporal resolution. We've used this method to achieve 16-second frame durations. We did play around frame durations down to 4 seconds, but we weren't convinced that we were seeing an identifiable signal. So we're now exploring whether improved reconstruction techniques can provide an even better temporal resolution. And we reported the results of some of this work in this paper by Sudishan in NeuroImage. So in summary, FPET allows measurement of glucose uptake with a temporal resolution that's within the realm of what we're used to fMRI, although much more work needs to be done to really get it to reach that point of fMRI temporal resolution. FPET provides the opportunity to measure a correlate of neuronal activity as biophysiologically closer to neuronal activity than the BOLD response. But the combination with BOLD also provides the opportunity to explore the fundamental bases of neurovascular coupling. I'm now going to briefly discuss one application of simultaneous fMRI-PET to resting state connectivity. And as with many topics in this talk, everything old is new again, and it's important to acknowledge that this concept of metabolic connectivity actually predates the concept of functional connectivity as measured with fMRI. Until now, the difficulty with these studies was the static approach, which meant that what we were actually looking at was covariance of the signal across subjects rather than connectivity within subjects. However, fMRI now provides the opportunity to examine this. So how do metabolic connectivity networks compare to those obtained using fMRI? So on the basis of static acquisitions, the literature has suggested that while there are similarities between FDG and BOLD resting state networks, there are also clear differences. And in particular, networks that bridge left-right homologue seem to be maintained, whereas those that cross the anterior-posterior dimension appear to differ or to be actually absent. And our paper in NeuroImage with 1 minute FPET resolution concurred with this hypothesis. The FPET and fMRI RSNs show clear similarities and differences. However, the reason for this remains to be explored. We've also examined how FPET connectomes compare to fMRI connectomes. And in this paper, we also examined static PET covariance matrices because we were interested to see whether or not the FPET approach actually does provide unique information that's not obtainable using standard methods. Static PET showed strong temporo-subcortical covariance both within and between networks, and strong fronto-subcortical, frontotemporal, and parieto-occipital covariance. FPET, however, was dominated by frontoparietal connections, both within and between networks. fMRI, on the other hand, showed strong connectivity within anatomical subdivisions, as well as a number of long-range connections between frontoparietal, parieto-occipital, and temporoparietal regions. Qualitative comparison of the networks graphs suggests that FPET seems to resemble fMRI more than it resembles SPET. And when we examined the shared variance between the three matrices, we found that FPET and fMRI had more shared variance than either static PET and functional PET or the static PET and fMRI matrices. And indeed, when we examined the region by region correlation between the matrices, we found that the static PET covariance matrix did not correlate with FPET above chance in any region. So the results concurred with our argument that an FPET is a more accurate representation of within subject metabolic connectivity than is static PET. It's important to note that direct comparison between these methods remains challenging because there are such substantial SNR and temporal resolution differences between the methods. It's also challenging because we should avoid using only one method, like fMRI, as the ground truth in the analysis. So it's quite easy to fall into the trap of using fMRI connectivity as the ground truth here because we know so much more about it. However, it's important to remember that each method should provide actually a unique but likely correlated index of neural activity. So work in this area continues. So to summarize, simultaneous MR-PET presents an excellent opportunity for exploring the fundamental mechanisms of human brain function. While some challenges remain, the technique is already providing new knowledge about human brain function, and I've presented an example of that with an application to resting state connectivity. To finish off, I'd just like to have a shameless plug of our work, where I'll highlight some of our recent work in metabolic connectivity, as well as papers where we've demonstrated the FPET method. We've also made our data available, and the resting state of data has been available for a few months now. And we recently made our checkerboard data available with full list mode PET data. And you can find these on Open Neuro. And I'll finish off with an acknowledgement of my collaborators and funding sources. Thank you very much.
Ladies and gentlemen, hello and welcome to my presentation about mapping task-based responses using simultaneous FPET fMRI. My name is Andreas Hahn and I'm from the Medical University of Vienna in Austria. Task responses can be readily identified in the brain with established MRI-based techniques such as BOLD and ASL imaging. And these are mostly related to cerebral blood flow. With PET, you also have the opportunity to image glucose metabolism, which is another essential aspect of neuronal activation. However, with the conventional bolus application of FDG, you can image task-specific effects only in the beginning of a scan, which has the disadvantage that several measurements are required. The solution to this problem is constant infusion, where FDG is then available for the actual metabolism at hand at any given time point. And any change in the metabolism will be reflected in a proportional change of the slope of the time activity curve. Once you have acquired your data, here is an overview of the FPET analysis. So you basically take your time activity curve here from the precentral gyros and your regressors from your design matrix, which include a baseline regressor as well as a regressor for eyes opening in this case and for right finger tapping here and here. And you feed all of this in a general linear model in order to separate baseline effects from task-specific effects. And finally, you can use the padlock plot for absolute quantification of the glucose metabolism. And this yields very nice task-specific effects in absolute units. Change is about 20% from baseline, so it's the method is sensitive, it's also regionally specific, and most importantly, you can acquire all of these things in a single PET scan. And this is the very basis for simultaneously mapping task responses with PET-MR. Of course, the method is subject to several assumptions. Some of them are that neuronal response and glucose metabolism are linked in a linear and time invariant manner. That's the same basically for fMRI, and that glucose metabolism is at steady state. We can further improve the FPET methodology, where it's very important to have a baseline regressor that is free of any task effects, otherwise the task estimate might not be accurately calculated. You can achieve this with different approaches. For instance, one can take the average of all gray matter voxels and fit this with a third order polynomial and exclude all task effects as shown before. Or one can use anatomical information and exclude those regions that are assumed to be active during a task. Or talking about simultaneous PET-MR imaging, one can estimate the individual fMRI activation and exclude all active task voxels. Furthermore, another improvement is the application of the radioligand in a bolus plus infusion protocol, which substantially increases the SNR, for instance, as you can see here, after 10 minutes of acquisition. And with this increased SNR, you can also decrease the task duration, for instance, from 10 minutes as we had before, to something which is probably ideal between 5 and 2 minutes, because otherwise it's get too noisy when the task duration gets too low. And this then yields a quite beautiful overlap between active regions identified in with glucose metabolism and bold imaging. We can also go a little bit more complex in terms of the actual task and the comparison across imaging modalities. For instance, in this study, we acquired simultaneously functional PET as well as bold imaging for connectivity and activation, and also arterial spin labeling data. And the task was a challenging visual spatial motor task, namely the video game Tetris at 2 different levels of difficulty. Just a word about the task design. In this case, we had a continuous task performance, which is quite feasible for the acquisition of ASL and connectivity, but you could also choose a hierarchical design, which is quite feasible for the acquisition of bold activation simultaneously. In this case, one would have slow blocks of task performance for the PET acquisition, and embedded in this is a fast block design for the fMRI acquisition. It just depends which kind of question you want to answer. So for this task, we also observed a substantial overlap between the different task responses, and on the right you can see the intersection across the 3 different imaging modalities. These task responses were also sensitive to subtle changes in task performance, for instance, when comparing the easy against the hard task conditions. And furthermore, we have recently shown that test-retest reliability of the functional PET method is quite nice, comparable to that of arterial spin labeling. It was a little bit lower for the bold responses, but this is in line with the recent meta-analysis. I would like to highlight that these different task responses represent complementary aspects of neuronal activation. The bold signal emerges from a differential increase between cerebral blood flow, which is high, and a lower increase in oxygen consumption. On the other hand, cerebral blood flow is mostly driven by glutamate release, and this will also increase the uptake of glucose into the cells to reverse the ion gradients. We can now use this complementary information in a framework of metabolic connectivity mapping, which is not to be confused with metabolic connectivity as mentioned in the previous talk. So MCM calculates the association between patterns of glucose metabolism and functional connectivity. First, one calculates the C2-voxel connectivity, which gives you the voxel-wise connectivity pattern in a certain brain region, and then this is combined with the pattern of glucose metabolism in a spatial correlation. And considering that the energy demands arise postsynaptically, in other words at the target region, the glucose metabolism enables to identify the target region and assign directionality to this connection. For the 3 regions and the task mentioned before, we calculated this directional connectivity in a collaborative effort with Australian researchers, and at resting state we observed a significant association only for the connection from the intraparietal sulcus to the frontal eye field, which was also stable for the 2 levels of task difficulty. For several of the other connections, we observed a significant increase in the association between glucose metabolism and functional connectivity when changing from resting state to task performance, and this was specific for feed-forward connections. And this made us to conclude that task-specific reconfiguration of functional brain networks carries a major metabolic cost. We then went on to investigate how such task effects will further change after prolonged training of the task. So we used the occipital cortex as a target region and observed that at rest, the association between glucose metabolism and functional connectivity increases in the training group, whereas during the task performance itself, it decreased. And simulation shows that the changes addressed were driven by changes in glucose metabolism, whereas the changes during the task performance were related to functional connectivity changes. So this suggests that metabolically cost-intensive connections are built by the training at resting state, and this then limits the required feedback during the actual task performance, which then results in an overall better performance. And for further details, please see the poster of my colleagues. Another interesting thing that you can investigate with this technique is the metabolic nature of bolt deactivations. This is work from colleagues from Sweden who performed a working memory task in a hierarchical design. And as shown before, the positive bolt response will also yield increases in glucose metabolism. However, in contrast to that, the bolt deactivations are not paralleled by decreases in glucose metabolism. In contrast, for some regions, the glucose metabolism actually increased. And this may indicate an active suppression of task irrelevant activity, which is metabolically intensive. And finally, I would like to show you that functional PET can also be used for the investigation of dopamine neurotransmission. So we can use the radioligand F-doper, which is specific for dopamine synthesis, because synthesis of dopamine is regulated by neuronal firing and also dopamine release. So during task-specific or neuronal stimulation, dopamine synthesis will increase, and this will also yield an increase in the radioligand uptake at the nerve terminal. So we have applied this technique in the established monetary incentive delay task, and this shows a substantial task-specific increase in dopamine synthesis in men for monetary gain, which was higher as compared to monetary loss in the ventral striatum. Interestingly, in women, we observed the exact opposite pattern, where dopamine synthesis was higher for loss than for monetary gain, and this was also significantly different between men and women. Furthermore, we could link this to behavioral data where dopamine synthesis changes were correlated with the reward sensitivity in men and punishment sensitivity in women. For direct comparison, we also assessed the fMRI response, which showed a robust activation of the ventral striatum for the contrast gain compared to loss, but this was not significantly different, not in this experiment nor in previous studies. So this indicates that the fMRI bolt response may not be equalized with the underlying dopamine action. So to summarize, I hope that I could show you that simultaneous FPET fMRI imaging enables the assessment of the task-specific effects in numerous different imaging modalities. These provide complementary information and also offer novel opportunities for different combinations. And you can also extend this for specific dopamine signaling. Finally, of course, FPET has some limitations as any other imaging modality. The biggest issue probably is the temporal resolution, because you need simply a certain amount of radioactive counts to create an image. But I think it's already quite good, quite a good increase from hours to days previously now to minutes or seconds. And the second issue is radiation exposure, but this can be further reduced with the next generation PET scanners. Finally, I would like to thank the team from the Medical University of Vienna and also our collaborators from Australia and Sweden, the funding institutions, and of course I would like to thank you for your attention, and please feel free to contact us if you have any further questions.
Hi, I'm excited to be part of this symposium at OHBM, and today I'm going to talk about dopaminergic mapping using simultaneous MRI-PET. We've already heard about very exciting developments in PET MRI, and in this talk, I'm going to focus on imaging receptor binding and neuroreceptor PET and fMRI imaging. Now, neuroreceptor imaging with PET has been done for decades, and it's traditionally being used for determining in vivo drug binding to a specific target. As you can see in this example here of the radiotracer F18 falipride, which binds to D2, D3 dopamine receptors. And it is shown in competition with increasing doses of the antipsychotic aripiprazole. So using this method, you can determine receptor occupancy for each dose of an antipsychotic drug in this case. Now, drug binding can also be evaluated using functional MRI. And of course, in fMRI, we think of imaging the brain's functional response due to any kind of stimulus on activation, but specifically when you're using a drug, we would also note this as pharmacological MRI, where we're looking at the in vivo drug response or the functional signaling due to the injection of a drug. And there are several studies that have shown that you can use this, for example, in this case, looking at the changes in cerebral blood flow due to amphetamine. And of course, with the introduction of simultaneous PET MRI methods and devices, this has become a really exciting area, because now what we can do is to simultaneously acquire PET and fMRI signals and evaluate the effect of any pharmacological agent or a drug, and really get simultaneous signals and information about receptor binding and very specific molecular signatures, and understand how, when a drug binds to a receptor, how that drug alters neuronal activity, which we then in turn expect to alter energy consumption and change the vascular response, which is something we can then pick up with fMRI. And so in the spirit of that, I'm going to present today a series of experiments, as well as some new results that fall into the line of exploring and understanding the relationship between receptor binding and a functional response. So first, I'm going to talk about how we've imaged the D2, D3 dopamine receptor system with the radiotracer C11 raclopride and a variety of pharmacological challenges in anesthetized non-human primates. And we've evaluated 3 different classes of drugs, antagonists, a partial agonist, as well as an agonist, both preclinically and clinically. And what we're using at MGH at the Martina Center is combined PET MRI, the brain PET imaging scanner. And you can see that in the study design, we truly acquire simultaneous fMRI and dynamic PET. The radiotracer is injected as a bolus infusion in these studies to allow maximum sensitivity. And we acquire functional MRI in the non-human primates with an iron oxide contrast agent to allow us to convert to cerebral blood volume changes for improved contrast to noise. And scans are on the order of 100 minutes for the overall duration. And through the middle of the scan, around 35 minutes, we would then inject a drug in order to evaluate both functional as well as dynamic PET changes. So here are the results that we get. What you see here is a baseline image of C11 raclopride, which shows very high binding of D2, D3 receptors in the striatum, which is really what we would expect with this radiotracer being sensitive to two subcortical measurements and the highest density of D2D3 receptors in these areas. So we then looked at the antipsychotic aripiprazole, and which is also known as a partial agonist, where we see a very high occupancy, really up to 92% change in binding potential of the D2D3 receptors. And at the same time, we see a slightly positive CBV response that's localized to the striatum, in this case, the putamen. Now, this is really mostly interesting when comparing this to a full antagonist. And what we find here in looking at two different full antagonists, raclopride and prochlorperazine, when there is very similar occupancies of 90% of the receptors due to a drug injection, we find that there is a very strongly positive localized CBV response to the striatum that we can detect using fMRI. And so both of these fall into a similar response, where we have an antagonist that's really giving on a full response, and a partial agonist or partial binding giving only a partial CBV response here. And then looking at a full agonist response, what we see here is that drug binding to D2 receptors on the order of 50 to 70% as an agonist. We now see corresponding CBV signal changes that are again localized to the striatum, but are now flipped in sign, and they are negative. And so it's really the difference in function that we can detect using fMRI here, where we get a negative response with an agonist, which is consistent with the fact that the D2 is an inhibitory receptor, and a positive response with an antagonist, because now there's competition with endogenous dopamine, and effectively something in between with a smaller response using a partial agonist. Now, that was an overview of the results, but we're also very interested in dynamic signatures of these signals. And what we find is that with the D2D3 antagonist, we see very similar temporal responses between PET and fMRI signals, which is concordant with a classical occupancy model. Whereas with the agonist, we find that temporal responses between PET and fMRI are diverging. So now PET-specific binding stays prolonged decrease. So if there's a prolonged occupancy increase, and the CBV response returns to baseline much faster compared to the PET response, which suggests that this signature indicates receptor internalization mechanisms. And this is what I want to go into in a little bit further detail with a continuing study here. So what is receptor desensitization and internalization in this context? It is a homeostatic neuroadaptation mechanism at the synapse that modulates cell signaling. And it's known to occur in response to G protein coupled receptor activation by agonist binding. When there is an agonist that's bound to a receptor, it can initially desensitize and then internalize, which can happen relatively fast, which can then lead to both short-term and long-term tolerance of the receptor. There is in vitro evidence that this occurs, specifically at dopamine receptors, but also at other G protein coupled receptors. And in vivo, it's not really known to be able to do a direct measurement of receptor desensitization and internalization, because and therefore its dynamics are unknown. Yet we do know that it does affect neuroimaging data, both fMRI and PET. So Being able to get a handle of this measurement non-invasively would be important. If we can do this with PET MRI, it would allow us to enable evaluation of several aspects of drug dynamics and receptor function or dysfunction in disease. So in vitro internalization of D2 receptors occurs relatively fast, on the order of 5 minutes. And so one of the questions is, what is the rate of internalization in this context, and can we measure that? So in order to do that, we looked at specifically amphetamine-induced dopamine release. And it is known that the release of dopamine following amphetamine administration is very long-lasting. But dopamine returns to baseline after about 2 to 3 hours. However, PET signals stay decreased for about 24 to 48 hours, which cannot be explained by just presynaptic dopamine release. Another piece of background information that's important here is that there is a couple of studies that have suggested that D2 receptors degrade in response to dopamine, but D1 receptors undergo different mechanisms and they recycle more quickly back to the surface membrane. And so keeping this in mind, what we did is a specific experimental design looking at repeated amphetamine injections. This was done in non-human primates, and we looked at IV amphetamine of 0.6 mg per kg. And this project was also led by Hannah Hansen here. So we start out with this initial first injection with amphetamine and simultaneous raclopride and fMRI signal acquisition. And we then repeat a second injection of amphetamine after either 3 hours or 24 hours in order to evaluate whether amphetamine can cause rapid desensitization internalization in vivo, but potentially could look at different recovery or externalization times. And in order to distinguish between D2 and D1 receptors, we also introduced a D1 receptor blocking in a separate set of studies looking at these results, looking at the same study design. And what we find is that with amphetamine, we get a response, we get receptor occupancy here that you can see. This is change in binding potential after both 0 and 3 and 24 hours. It's most pronounced at 0 hours and a little slightly less at 3 and 24 hours, but mostly similar. What's interesting is the corresponding cerebral blood volume measurements, which are shown here, both positive and negative signals. At 0 hour, we get a predominantly negative signal, whereas at 3 and 24 hours, we now get a flip in sign. We get a positive response at 3 hours and somewhat of a mix after 24 hours. We can see this in more detail in these time activity curves, where you can see the 0, 3, and 24 hours here in the putamen. And you can see that binding potential and as well as occupancy, binding potential decreases and occupancy is elevated to on the order of 20 to 30% using this amphetamine injection. And it slightly goes down after 3 and 24 hours. Now, the CBV response in the putamen is initially negative at 0 hours, but then flips to positive and goes back to a predominantly negative, but a mixed response after 24 hours. As I said before, we then looked at what happens when you block D1 receptors using sharing 23390. And what we see here is most interestingly that the response at 3 hours, the CBV response at 3 hours now is not positive anymore, but is really diminished, when blocking D1 receptors. And we can see this similarly in binding potential changes as well as occupancy, as well as on these maps here when we now look at the same set of experiments when there is a sharing blockage before administering amphetamine. We see that the response at 3 hours now gets eliminated compared to not blocking D1 receptors, suggesting that this response with just amphetamine at 3 and 24 hours, that the positive response is really driven by D1 receptors. There is also an interesting piece of really signals here where we see with just amphetamine, we see mainly a change in caudate nucleus accumbens that is consistent, but in the thalamus, we see a slightly positive response at 3 hours. That is then changed when you're introducing sharing as a blocker before the amphetamine response. So to conclude, we've looked at repeated amphetamine exposure that caused continuing decreases in C11 raclopride binding, which likely represents a combination of dopamine release and receptor internalization. And the negative CV response after the first amphetamine injection suggests that D2, D3 receptors really dominate the 0-hour response, but that D1 receptors remain available and dominate the response after 3 hours, suggesting different recycling rates between D1 versus D2 receptors. And so overall, I hope that you find this interesting piece of information as an example of how we can use combined PET and fMRI signals to understand receptor signaling mechanism in much more detail. With that, I'd like to thank funding sources as well as all the people and collaborators involved in this study. And thank you very much to the organization of the symposium.
Good morning, good afternoon, or good evening, ladies and gentlemen. It's my pleasure to present some thoughts and results on trimodal MR-PET EEG and close my presentation with some discussion about future directions for MR-PET at even higher fields, such as 7 Tesla. Let's think carefully about the advantages of each technique and see if it's worth bringing them together. This fingerprint diagram shows some chosen attributes that can be applied to any imaging technique that have been chosen to illustrate the points I want to make. Let's start at the origin. The further out we go along any axis, the better that particular attribute is fulfilled. MRI is the method of choice for in vivo anatomical depiction, and you'll see it's been given a rating of excellent for anatomical depiction. It's also pretty good at showing function, as in fMRI, that's why we're all here. If you look at its spatial resolution, which obviously correlates somewhat with an anatomical depiction, it's excellent, can go down to 0.5 millimeter easily these days. One can find MRI in most hospitals. Its clinical use is excellent, as is its clinical availability. Technically, it's a very robust instrument, but surprisingly, even for a method that's been around for 30 years, its developmental prospects are still quite remarkable. 7 Tesla and higher fields are being explored. So this fingerprint diagram represents its strengths, but if you look at the weaknesses, temporal resolution is not great. Molecular imaging and specificity and sensitivity are also very, very poor. If we now look at the same attributes, but for PET, you'll see that PET's not great at depicting anatomy. Functional, it's great. You can choose which tracer you use. Temporal resolution is also poor. Spatial resolution is not great. But if you look at molecular imaging and the sensitivity and specificity, it's excellent. PET can be up to 6 orders of magnitude better in terms of its sensitivity than MRI. And of course, it's very, very specific depending on the tracer you use. Clinical use is great, but unfortunately, it's not available in too many clinics. If you're fortunate enough to be able to combine these two instruments in one machine, you'd have most bases covered if you could do simultaneous measurements using MRI and PET. But again, temporal resolution would still escape you. And if you were then able to combine EEG in the same scanner, same recording, and do these three simultaneously, you'd have all of these chosen bases covered. And that's one of the reasons we have been trying to push towards MR-PET EEG. That's great looked at from the technical point of view, but what does it bring us in terms of real functionality? Recently, the National Institutes of Mental Health in the USA proposed the use of research domain criteria that make no assumption about current symptom-based categories. These criteria build a framework for studying mental illness across basic dimensions of mental functioning, such as emotion and cognition, cutting across traditional diagnostic categories. They encompass multiple levels of analyses, from genes to neural circuits to behavior such as fear, executive function, working memory. Thus, having multimodal data will surely make data-driven categorization more useful and lead to a more precise diagnosis. Now let's have a look at some biomarkers for brain disorders. This slide shows an example of how functional imaging abnormalities may relate to neurochemical abnormalities and candidate genes, and also to psychopathological key components of brain disorders. For the purpose of clarity, this figure does not indicate all functional directions, but the connections between the layers are evident. I hope the advantages of the trimodal approach now become clear as the biomarkers now segregate out into different modalities, as clearly depicted by these lines. At this juncture, I'd like to give a quick introduction to the concepts of EEG microstates, whereby a number of approaches have been proposed to extract features of neuropsychological relevance from EEG recordings. Microstate analysis is one such method that defines states of the multi-channel EEG signal by spatial topographies of the electrical potentials over the electrode arrays. Basically, this is a condensed framework linking measured electrical potentials and topographies which can be investigated further. For illustrative purposes, this figure presents a 12-second recording, resting state recording, of EEG with eyes closed, showing a period of strong alpha activity lasting about 1 second. For a closer look, here's an expanded segment of the EEG activity during an alpha burst. Electrical potential maps at successive points of the global field power peak during the 2-second EEG are shown in figure B. Periods of stable map topography with different durations become apparent. These are marked in different colors, which are also superimposed on the EEG in figure B. The spatial distribution of active neuronal sites that contribute to electrical activity in the brain can be revealed using EEG scalp topographies. Such topographies are found to be quasi-stable for periods of about 80 to 120 milliseconds. Each of these topographies is then called a microstate map, or just simply a microstate. The functional interpretation of microstate maps assumes that each map or topography is generated by diverse neuronal assemblies. Their locations can be estimated using source localization techniques such as standard low-resolution brain electrode tomography, the so-called Loretta method. The transition of microstate maps may be regarded as sequential activation of various neuronal assemblies. The global field power, the GFP, shown in red in this slide, can be calculated as indicated at each instant of the multi-channel EEG recording. Peaks of the GFP curve represent moments of highest signal to noise. At the peaks of the GFP curve, the potential recorded at each electrode of the multi-channel signal is plotted onto a map of the channel array. This collection of maps is entered into a clustering algorithm, TAAHC or K-means clustering, which results in a small number of representative microstate maps that explain a large proportion of the global topographic variance. 4 topographies are repeatedly found using this method. These methods are simply labeled A, B, C, D as shown in this figure. The original maps of the peaks of the GFP curve are assigned to a microstate class A, B, C, or D based on the degree of correlation with the microstate maps and statistical smoothing of the time series. This reassignment results in a representation of the original multi-channel data as an alternating series of microstates A, B, C, and D, as shown in the bottom of the figure. Numerous resting state EEG studies have consistently identified 4 topographies. A collection of results with the corresponding topographies is shown in this slide. The similarities of the states between the studies is, I think, evident. Now let's have a look at the advantages of this multimodal setup. Precision medicine clinical data can be combined with patient information, a more targeted approach to brain disorders, individual assessment, including treatment and diagnosis, an interdisciplinary approach given that we're using a number of different modalities as well as in vivo biomarkers, leading to multimodal fingerprints, and of course then these can be used to look at brain disorders. It would be somewhat remiss of me not to mention the problems involved in integrating these different modalities. Let's have a look at what happens when you integrate EEG into an MRI machine. There are a prominent number of MRI-related artifacts in EEG recordings. They result largely from the switching of the gradients and pulsing the radio frequency. As shown here, these are EEG recordings which have been completely wiped out by the gradient switching, but they can be cleaned up. And then we have to deal with cardiac-related artifacts, the so-called cardioballistic artifact, as shown here. Each time the heart pulse, there's movement, and that can be seen in the EEG. As shown in this slide, this is the cardioballistic artifact recorded in an ECG at 0 Tesla, 4 Tesla, 7 Tesla, and 9.4 Tesla. And you can see without any switching of gradients, just simply because of the static field, this is getting successively worse as you go towards higher field. This can be cleaned up, thankfully. And then there are other motion-related artifacts, movement of the conductive materials, the electrodes, displacement of the head in the magnetic field, vibration induced by the helium pump, gradient switching as well, and then endogenous and inevitable other motion that takes place. Let's have a look at the trimodal setup of MR-PET EEG. fMRI EEG integration takes place like this. The MRI console is shown here together with the EEG monitor, and these are all linked by having a synchronized clock shown here. And then the EEG system goes inside the MRI scanner. Short electrode cable length is advised. Fiber optic cable goes from the box to the outside world. And as I already mentioned, clock synchronization is really important. Then in the trimodal setup, it works like this. So we have the PET data acquisition. The example given here is FDG. The experiment starts with an injection of FDG, approximately 200 megabecquerel, and the experiment lasts about 60 minutes, during which dynamic FET recording takes place. The MRI data are recorded also during this time, and of course you're at liberty to decide which MRI sequences you need to use, such as structural or functional acquisitions, and then during this time the EEG data are also recorded. A typical protocol might look like this. MRI normally begins with an MP-RAGE sequence and then followed by tetrastar weighted EPI for the functional imaging, for example. The PET acquisition will run in list mode. The data are subsequently corrected using MRI or a template-based attenuation correction method. The EEG, in this case a 32-channel system that we were using, sampling rate of 5,000 Hertz, bandwidth of between 0.016 and 250 Hertz. Data, looking at some results now, a comparison of bold and glucose consumption within the default mode network. On the left, you have the fMRI results. On the right, we have the PET results. One can see the similarities very, very clearly. And below, we have the mean activity of the bold in the default mode network and the non-default mode network, and correspondingly on the right, the normalized SUV values from PET for the default mode network and also the non-default mode network. Okay, let's have a look at those results in a little bit more detail. Let's explore the correlation between the mean fMRI bold and the PET SUV values. As shown in this diagram, and clearly they correlate quite nicely. And of course, on top of that, you have the EEG results, a depiction of the EEG results of the alpha band as processed with S-Loretta. And of course, one can dig deeper into the EEG results and look at the different bands, alpha, theta, delta shown here, for example, on the top row, beta 1, 2, 3 in the bottom row. Studies that we've looked at excitatory inhibitory balance within EEG microstates and resting state networks. This experiment takes place in the following way. It's a bit more complicated. PET acquisition begins with a C11 tracer, ABP. Also the MRI data running simultaneously with the EEG, in this case 64-channel recording. And then to look at the GABA, F11 flumazenil PET is run again together with the fMRI acquisitions, and you can see here now structural MRI spectroscopy, fMRI resting state, auditory stimulation resting state again, followed by spectroscopy, and the 64-channel EEG recording runs from when the fMRI starts. EEG results then in these microstates as described earlier. And of course, the usual measures from fMRI can be depicted, and SUV values from PET of the FDG tracer, of the ABP tracer, and of the flumazenil are shown here in this slide. And all these can be brought together to produce the multimodal fingerprints that we talked about. So briefly, I'd like to close my presentation by talking about a few future directions for MR-PET at ultra-high field, for example, 7 Tesla. Simultaneous MR-PET imaging of second generation is a project that we've been carrying out for a number of years to improve the resolution of the PET instrument and make it compatible for 7 Tesla. This slide depicts the major components. The PET insert, again, only for brain imaging, goes inside the bore of the 7 Tesla scanner, as depicted also here. And the gantry that holds the PET cassettes, shown here. The PET cassettes are in this outer ring, and an RF coil goes then inside the PET ring. It's important to note that the MRI coil has been designed and built with PET in mind to have as few lumped elements in the PET field of view as possible. The improvement in the resolution of the PET comes from choosing a multi-layer design as depicted here and here, 3 staggered layers with some displacements to give us the improved resolution. And if we look a little bit more carefully at that, the first layer, second layer, and third layer then give you points in this way, and this leads to much improved resolution to give some depth of interaction. And this is an actual picture of the stack itself with the silicon photomultiplier tubes glued onto the bottom. The silicon photomultiplier tubes are, of course, MR insensitive, and these are the so-called flood maps showing you that the resolution is indeed improved. Looking then at the ultra high field compatibility of the PET cassettes, carbon fiber housing to keep all the electrical noise generated inside those cassettes, cooled with water, the acquisition electronics consisting of digital silicon PMs, the stack also mounted as shown here to give full field of view brain coverage, and with heat sinks to reduce the thermal variations in the setup. As I mentioned already, those cassettes, 32 of them, slot inside this gantry, and the gantry then goes inside the MR scanner. The gantry is open at the back to allow for any screens that one might use for neuroscience studies to be viewed, with a small mirror because space is a premium once the head coil goes in. And obviously the isocenter of the MR and PET are coincident. And there's a little bar here that allows for accurate repositioning between studies. I would like to emphasize this project that's been going on for a couple of years. We're coming to the end of it, and I think by certainly human brain mapping next year, we'll have first results with hopefully a PET resolution of about 1.6 to 2 millimeters isotropic over the whole brain, including the cerebellum, and we look forward to being able to present the results to you. I'd like to conclude by thanking my colleagues who've contributed to this work and also to acknowledge our funders. And thank you very much for your attention.